This recorded session is the second half of our module on microbial communities and the factors affecting their assembly. So, I think we ended with the discussion on microbial community structure and microbial succession, the factors affecting it. In particular, we have already discussed how broad scale and fine scale competition affects microbial community succession. So we can think of competition as a major factor influencing or shaping microbial communities. So initially, many species will colonize a new habitat. So after a disturbance, we expect multiple species who will try to or which will try to establish themselves in a new habitat. And as we can see here, there's an increase in their population. However, the competition as a factor will soon arise. Broad scale resources will of course be resources where competition will still exist. So you expect a number of species of a particular microbe to decrease as such kind of competition exists. And initially, the structure of the community at the onset of succession will be greatly influenced by broad scale competition. We expect niche spaces to increase. Um, so there will be more specialists that will be capable of establishing themselves. A fine scale competition comes into play. So fine scale competition will succeed broad scale competition, as fine scale competition increasingly structures the niche space of the habitat. The number of the species again will definitely increase. So you can see here, in the sigmoidal curve, signifying the increase and decrease in the population number or the population or the microbial the size, of a specific microbial species. So broad scale and fine scale competition actually are factors that succeed one another, and they're very important in structuring microbial community. So, as mentioned, um, these two types of competition, they follow a sequence, and they will definitely affect how the community will progress. As species colonize new habitats, the richness of that habitat will increase over time. You expect communities or multiple species to aggregate with one another for forming biofilms. No? Along with the increase in species richness, resource diversity and niche space increases, as I've already discussed, no? Yung early part on broad scale competition. The microorganisms colonizing the new habitat will definitely alter the environment. No? So eventually, when the microbial communities become very established in their new habitat, that's the time that they will now structure the physical and chemical status of the environment. They will create new niche spaces that can be colonized by other specialist microbes, and of course, no, as microbial communities, the members of the microbial communities, as they interact, they will definitely produce waste products that may actually support additional microbial species, especially those that thrive on waste material as their primary, I don't know, organic sources. So niche space will increase over time as microbial communities become more established, they become more prominent. So makakakita tayo ng mga microcolonies on the different spaces or surfaces within the colonized habitat. No? So, okay, so I've mentioned this already. And then also we've discussed about fine scale competition, the succession between these two types of competition. Okay. Yeah. Okay. So moving forward, so we've established, or the microbial communities already established themselves. After a while, they will develop or achieve a sense of stability, a sense of stationary existence, or uh, stationary coexistence with one another. So hindi siya problematic. That even though you have multiple species in a microbial community, since they have different resource needs, they have explored different niche spaces, and as long as the resources are not depleted, no, in a natural environment, we expect the constant flux of resources, no. But also replenishment, kung ano naman siya, it's a newly or it's a habitat that's beginning to to flourish. We expect replenishments of the nutrients. No. Over time, the microbial communities as biofilms on that new habitat, they will mature. The community will become more structured. Microbial species will have dedicated niche spaces, dedicated resources that only they can actually utilize. No? And eventually, after a period of stable or stationary coexistence, the community can be restructured again. But this time, instead of the microbes actually doing the restructuring, it's a third party doing it. So we have predatory organisms. Those organisms that feed on microbes, sila naman na yung next factor or next player that can structure the microbial community. So the more that they eat a specific species, the more that microbial species will actually be will actually be eliminated. No? And the more that others are capable of veering away or camouflaging themselves para hindi sila ma-detect ng mga predatory organisms like viruses, protists, and other more mga predators or mga filter feeders. Those organisms naman, those microbes that have the capacity or that develop the response of camouflaging or being not detected, nakaka-survive sila from these from the predatory element or the predatory predation factor that actually structures a microbial community that already achieved a certain degree of stable coexistence. No? Okay, so now that we've really discussed about how factors such as competition, predation influences microbial community structure, let's dig deeper into the concept of microbial communities. No, we'll introduce the idea of species diversity. When we talk about species diversity, we actually talk about multiple concepts. We include the number of the species found in the community. We call that factor or that concept the species richness within the community. We also talk about relative abundance or evenness. So in a particular environment, we expect that the species within communities, they have a particular number, no, or a particular microbial size. So their relative abundance factored in is actually a concept of species diversity. The thing with species diversity, when we talk about microbes, is it's quite complicated. Kasi kung i-compare natin siya sa species concept ng vertebrates, ng animals and plants, remember that in bacteria and in archaea, in the domain domain eubacteria, okay, and domain archaea, the definition of species is very dynamic, and in fact, until now, we really have I don't know, we really have some debatable narratives or some elements that are debatable. Say in bacteria and in archaea, we consider a species to be a specific one or to be a particular one if let's say, for example, no, from an environment we collected two colonies, and we were trying to figure out if they are actually belonging to the same species. They look the same when we grow them in the lab, in vitro, and we wanted to know if they are actually belonging in the same under the same species identity, under the same species ano no tax what they call the species taxon. So they may look the same in the outside, but to verify their identities, we I don't know we factor also molecular data. We factor molecular identification as an element to actually discuss more about species diversity. We have this so-called DNA hybridization. So in this technique, we isolate the DNA of the microbes that we want to compare. And then if 70% of their DNA strands hybridize with one another, we can consider them as belonging under the same species. Though the value, the threshold value is quite debatable pa rin. There are some who will regard or will require a higher degree of DNA hybridization before they consider two organisms, two microbes to actually belong under the same species. Also, we have biomarkers, no? Hindi na to news sa inyong lima. We've discussed heavily on on molecular identification and the use of particular genes of bacteria in order to identify them. In specifically, I'm talking about the 16S rRNA gene. So such biomarker, we amplify that from our from our microbe of interest. We sequence the gene, the amplified gene, and then we compare it using a database. We try to search in the database which sequence matches yung gene, yung 16S gene na pini-CR natin, and we have different cutoff or threshold values, no. Some authors agree with a 95% cutoff, others agree with 97, others require greater than that, mga 99, no, before they really believe na the organism that you have identified based on the 16S biomarker is actually that organism, no, in reference to a database entry. So, because of a lot of complexities about defining what a prokaryotic species is, and that actually factors in to metrics of a species diversity within microbial communities found in natural and artificial environment. So you have to remember that. Mas komplikado ang pagde-define natin ng isang species pagdating sa microbes, pagdating sa bacteria and archaea compared to plants and animals. Okay? Now, why should we actually concern ourselves? What's the need, no, for concerning ourselves in studying to study species diversity? Time and again, no, we have been talking about microbial ecology as something, or we've been regarding our study of microbial ecology as something that's shifting from a framework that used to depend on successful isolation of colonies to a newer paradigm that requires only mga biomolecules like nucleic acid in order to describe the identity or to give an identity to a specific microbe. So the concept of the Great Plate Count anomaly is the reason behind that, because we can really say, or there are a lot of attempts already describe the phenomenon na we can really grow a lot of microbes that we get from the environment. We can really replicate their growth in the lab. So we essentially have what we call an uncultured microbial majority, and that population represents a very potential filled set of living organisms that we may harness in the future yung kanilang mga mga metabolic capacity, so yung kanilang yung kanilang natural tendencies we may be able to harness in the future, or on the basic research part or the basic research dimension, themselves alone, as we study them, of course, that will help us understand our environment more, no? Since microbes are the primary architects of physiological processes that on a biosphere level, on an ecosystem level, we know for a fact that microbes are the primary architects. Okay, so such processes that I've been emphasizing include our elemental cycles, mga biogeochemical cycles, and there are microbe facilitated chemical transformations. So the more that we know microbes that are, or the more that we discover the diversity of microbes that can carry out such chemical transformations, of course, the more that we understand about how these elements actually cycle. Also, we have the process of decomposition. Diba? To give an example, meron tayong mga interests on microbes that can degrade plastics, microbes that can degrade oil, no? What else? Microbes that can, what do you call this, that can help in the decomposition of organic matter that's hard to really decompose. Ayan, no? So, decomposition is a natural process, very much involved or participated by the plethora of microbes that are found in our environment. Then, of course, on the human health side, yan, we also want to know, or we study species diversity, especially of pathogenic bacteria, because they're causative agents of human diseases. And the more that we know about them, the more that we can plan for novel treatments and managements to resolve cases arising from human infections caused by these microbes, by these pathogenic microbes. No? Okay. Now, how do we study species diversity? There are a wide array of tools that are already existing in order for us to measure the diversity of I don't know of macroorganisms of plants and animals. And as we proceed to also studying the diversity of microbes in our planet, the applicability of these tools we've also explored definitely. No? In fact, yung mga concepts natin ng species diversity sa microbial communities, heavily lifted din sila, no, sa ecology ng mga macroorganisms. Some examples of those indices, those tool set or those elements of diversity tool set that we use to study species diversity include those indices that are indicative of species richness, the number of species that are present in the community, indices of species evenness. So here we factor proportional or we factor abundance as an element. If we want to try to articulate or compute for the evenness of species, so that we can have a picture of how the microbial community actually looks like, what dominant species are found on top of the food pyramid within a microbial community. And we also have within the idea of evenness, how species abundance is actually distributed. No, na are there pockets are there pockets within a natural environment where you expect a certain species of microbe to occupy? Are there like pockets of niche spaces na kung saan only a specific microbe or group of microbes can occupy because they're highly adapted in dominating or colonizing such particular environment? So those these indices are useful in the study not only of species diversity in animal and plant communities, but also in microbial communities. To give some examples, so we have our Shannon Index, the Shannon-Weiner Diversity Index, and the Simpson Index. So let's go ahead and discuss these two indices. Let's begin with the Shannon Index. The Shannon Index, it is a measure of the degree of uncertainty in predicting the identity of the next individual that is sampled in a community of S species and N individuals. So the Shannon Index, very useful in you know, really telling or really elucidating how diverse the microbial community is. Now, how do we, what things actually affect this value, this index? The Shannon Index is influenced by the number of rare versus dominant species in a community. Primarily, it gives weight to the rare ones, no? To more rarely seen or rarely identified species. Normally, if you did not sample properly or hindi ka nagkaroon ng representative sample within an environment, that's the time that your Shannon value, that your Shannon-Weiner diversity value, it will be useless. It will be just an underestimate. No? So, important that if we are trying to capture species diversity of microbial communities within an environment, always remember that you sample following systematic methods, no, in order to ensure that you have a representative material or a material representative of the environment where you will isolate your microbial communities. Okay, essentially, whatever you extract later, whatever data you produce later, ano siya, applicable siya sa kabuuan ng environment. So, very important ang coverage. In general, yun nga, no, na-mention ka na, aside from affecting yung, yung computation of the Shannon Index, a coverage that is not, what do you call this, hindi properly executed, a sampling that's not properly executed will definitely influence also evenness, 'di ba? Hindi lang yung ating diversity index na Shannon-Weiner. The evenness of the microbial species within the community is also influenced. Okay? How do we compute for the coverage of the sample, and how do we ensure na na maganda yung natin, or malalim yung ating covered na sequencing depth? That's something that we will cover in the laboratory, primarily using the Mothur pipeline. But before, marami or early forms like the Dutour, also of the same same author. That's one pipeline that we use to check on the coverage of our sampling. Next, let's see. Next thing we have to discuss is the Simpson Index. No? But for the Simpson Index, we define it as a measure of diversity, which takes into account the number of species present, not only yung type, no, or yung nature of species, but the number and essentially the relative abundance of each of these species. As species richness and evenness increase, we expect the diversity will actually increase. With Simpson Index, a valued or an index value that is equivalent to 1 represents infinite, very infinite diversity, while the more closer the value of the Simpson Index to 0, the more na walang diversity or mababa yung species diversity in terms of abundance, in terms of richness. I think I have here a video discussing Simpson Index. Let's watch this.
The equation for the Simpson's index is this. D equals 1 minus the sum of lowercase n multiplied by n minus 1 divided by capital N times n minus 1. The variables in this equation are D, which is the diversity index, lowercase n, which represents the number of individuals in a single species, and capital N, which represents the number of individuals in the entire population. Our sample problem is going to contain 5 species. If you want to find the Simpsons index for this population, the first step is going to be finding the value of the numerator. The easiest way to break this down is imagining that the numerator, n times n minus 1, is its own equation. You need to input the population values for each species and then find the sum of each of these values. In our example, the numerator would look something like this: 13 times 13 minus 1 plus 42 times 42 minus 1, etc. After you input all of the values, your equation should look something like this. Next, you need to find the value of the denominator. To do this, you need to take the total population number and input it into capital N. A quick side note, I've taken the population values that we calculated in the previous step and added them together to find a total of 2,612. Now, our denominator using the capital N of our sample population, which is 100, should equal to 100 times 100 minus 1, or 9,900. Now, all that's left is to solve for the index value. After completing your calculation, you should get 0.74. Remember that the closer to 1 your index value is, the more diverse the population is. This means that our sample population is pretty diverse.
Okay, so that's a very nice, a very simple, representation or discussion about how we get the Simpson index. Probably you've encountered Simpson index computation in your macroecology, no? Yung ecology ninyo ng second year or first year. So in our case, for our case, the Simpson index computation, we will actually follow or we will get it following the pipeline, the data analysis natin using sequence reads. So it's something na it can be, I mean, I might be able to, I may give problems like that, but for in practice, we don't calculate the Simpsons index manually anymore like that. Kasi for our case, we're dealing with thousands to hundreds of thousands of I don't know data points. So it's impractical to do that. Pero just to just for you to have a clear grasp of how or what the Simpson index is. Yan. That's a very nice video discussion. Okay, so we're done with Shannon Diversity Index and Simpson Diversity Index. So just remember the difference between them. Okay, that in your Shannon index, the primary the primary thing that influences the value is the the number of rare species versus the number of dominant ones. While for Simpson index, we factor relative abundance. Okay, now we have to we factor relative abundance as, and then essentially we treat the community as one whole unit to be analyzed. Okay, so now let's move forward to another concept. We relate to microbial communities and their assembly. We introduced the idea of microbial co-occurrence. Now, a key question that helps us understand how communities are structured and help us discover if there are actually some sort of rules or things that are followed, things that govern the structuring of microbial assemblies is this question, no? That are there patterns of co-occurrence that are randomly controlled, or are they affected by the environment, no? So yung ano natin, do we leave it or does this chance or randomness influence how microbial species co-occur with one another, or is it the environment with its biotic and abiotic dimensions affecting microbial co-occurrence? Is it are these patterns of co-occurrence influenced by competitive strategies among microbial communities? Are they influenced by cooperative strategies, or are they influenced by both? No. Now, in the study of macroecology, we expect na very heavy na yung nalalaman natin, no, about organismal co-occurrence. No? Now, the rules in organismal co-occurrence, does it apply with microorganisms that we are invested on, no, in our course. Okay, so um, in still in talking about species co-occurrence, it's been studied by multiple individuals, by multiple groups of microbial ecologists, and they found out that similar with macroorganisms, microbes as well, no, they show a great similarity in terms of pattern of co-occurrence. Like they did statistical comparisons with a null model that predicts that the patterns are actually just created by chance. So the experimental group or the experimental unit that's being analyzed, nakita that we actually see that species have what we call microbial species have what we call permissible combinations, no? That they can co-occur depending on their nature, depending on how they I don't know carry out everyday physiological processes. So we expect that the nature of microbes of microbial species actually influence the ano no the permissible combinations that they can produce with one another. So microbial co-occurrence is a thing. It happens, and it's another I don't know. It's another interesting avenue that we can, or another interesting what do you call this? A research question or research area in the study of microbial ecology, in the study of microbial communities. Okay, now we're nearing the last part of our discussion. We have the concept of disturbance in microbial community assembly. No? Let's talk about disturbance. Disturbance, it may take many different forms, and as I've mentioned in our synchronous session last time, it's an important element that actually shapes and affects colonization and microbial succession. No? So, aside from affecting, I don't know, the patterns of species diversity or in diversity itself within communities, um, several things can also be dictated by disturbance, like the nature of early colonizers, the physical and chemical what do you call this makeup of the environment will greatly be influenced, will greatly be altered because of disturbance. Some examples pala no of disturbances that can reset yung mga natural environments natin. Fire, hurricanes, and deforestation for natural environments. For the case of humans, disease can actually disturb our normal microbial communities. Like, let's say, no, nag-i-intake ka ng merong pinuscribe sa'yo na antimicrobial treatment. So, essentially, kung lalo na kung yun ay ano, no, GIT ang kanyang passage sa tiyan. Such antimicrobials can wipe yung ano mo normal microbiota mo on your intestines, and essentially it's a form of disturbance. So it will influence how succession and community microbial community restructuring will occur. Okay. We've already defined it, the process or the phenomenon of disturbance as an event that will cause the death, the decimation of life, or displacement to individuals within a population, and that will definitely lead to colonization or recolonization. Some as the examples that we have shown or that we have enumerated a while ago, let's take a look at some of them and their relationship to how community restructuring occurs. For the case of hurricanes, they are actually a big type or a large scale form of disturbance event. One study used the occurrence of Hurricane Frances as a natural experiment to study the effects of disturbance on cyanobacterial mats. The mat community that is targeted in this under this study appeared to be in an equilibrium state before the introduction of the hurricane. And changes in the community composition, and salinity in the equilibrium state did not change some ecosystem parameters like carbon nitrogen ratio. However, it greatly influenced the hurricane magnitude, greatly influenced or the salinity values by lowering them, and this resulted to deposition of sand layers on the cyanobacterial mat communities that actually resulted to shifts in community composition and carbon nitrogen ratios that destabilize, no? Essentially nag-cause ng disbalance ng equilibrium, yung kanina nga describe ko as stable coexistence. Dominant cyanobacterial species decreased, and more rare species existing prior the disturbance event, the hurricane, increased substantially in terms of proportion. No? So as an example, we can see hurricanes as very, very destructive, talagang dramatic yung effect on the community structure and also the status, the physical and chemical status of the natural environment. Okay? Aside from affecting community structure and the physical and chemical status of a natural environment, disturbances can also affect social traits such as cooperation and contention that hopefully we expect naman, no, in any form of microbial succession, the goal is always to coexist. So we expect that eventually, kahit na after a great magnitude of disturbance, colonization, and then microbial interactions with microbe interactions will influence or lead to stable coexistence. For the case of disturbance that are of low rates, no, although cell densities reach critical levels for cooperation to be beneficial, some microbial species that cheat arise, and they gain a selective advantage, eventually dominating the population. So in instances where the disturbance is not that high in terms of magnitude, yeah, no, there could be some microbial species that can take advantage of the event. They may be they may be selected for that kind of pressure, and essentially they can dominate. In on the other end naman ng ating spectrum, at high disturbance rates, those microbial cheaters, they predominate because of mass mortality events that prevent cell density from reaching a level in which cooperation is beneficial. By the way, ang context ng microbial cheater natin is a species that's not really cooperative of others. A species that will, probably wala siyang, hindi siya capable of production on its own, tapos gumagamit lang siya ng mga pre-made na nutrients. So, wala siyang ambag. No, yung mga ganoong microbial species, if they're selected for a particular disturbance, be it of high rate or low rate, they may take advantage of that disturbance in order to rise to the top, in order to be the predominating microbe. Okay? Now, we're at the last part of our discussion. We are now focusing on microbial community assembly. No? So far, we've discussed a lot about species diversity and microbial succession, factors affecting microbial community structures, eventually or essentially also affecting succession patterns. Now is the time for us to discuss the basic processes of microbial community assembly. So from the concept, from the conceptual synthesis by a renowned author on microbial ecology by Velend, there are four basic processes of microbial community assembly. These are diversification, dispersal, selection, and drift. So we'll be discussing each of these four basic processes. Let's start with diversification. So let's start with hindi ko nailagay pala ang diversification. Okay, anyway. So let's start with diversification. So diversification, actually, it's a central, it's a central process for microbial communities to be able to assemble. So when we talk about diversification, factor natin yung concept ng prokaryotic species. So diversification uh, uh, is associated with um, organisms, microbial organisms being able to, or given the shown to or observed to possess flexibilities that are given to them or that they acquire through time. They become more diversified. Their metabolic capacities become more diversified, or in some instances nagpo-form sila ng mga tinatawag natin na ecotypes. Like, under the same species, there are multiple ecotypes, multiple species that belong or that are found within the same environment but are able to exploit specific niche space. Tapos, on that niche space, sila lang yung nabubuhay. So, particular ecotypes. No? Now, as an element of microbial community assembly, of course, the more diverse the environment, the microbial community is, the more diverse the members are, the more na maaapektuhan yung mga factors like competition and predation. And essentially, diversity can either diversity will establish the assembly of the community, or it will demolish the assembly because hindi beneficial yung pagkakaroon ng diverse types or diverse level, very high diversity levels on an environment. Let's say if the environment is resource poor, maaring problematic if you have very high initial diversity, or if the microbial species dominating tend to diversify through time. So, two-prong yung effect ng diversification depending on the context of the environment. Then we also have dispersal. Unlike diversification, dispersal it has been studied less, primarily because of the fact that microbes have very small size, and to grow them or to observe them in natural setting is very challenging because they have short generation time. Now, as a proxy for dispersal, we use distribution. We use distribution as a indicator of microbial dispersal. Now, the rates and order in which taxa are added to a community, that actually varies depending on the context or depending on the natural environment. So, no two different environments will have the same dispersal pattern, will have the same microbial dispersal patterns. It will really be highly specific or highly unique, Even though sabihin natin parehas silang forest, the two different forests, pero still they are classified as a forest, the process of microbial dispersal on communities, let's say on the soil of the forest, can on those two forests will be unique depending on the entire nature of the environment. Okay. Then we also have selection. Selection significantly shapes microbial community assembly. Okay? Different habitats, they harbor different sets of microbes. Environmental factors such as pH, salinity, and carbon availability, they are known to affect structure and assembly of bacterial communities. Again, we're going back to our debate about biogeography and cosmopolitanism. I hope someday we'll be able to answer the question, but it's really probably will take more time. Kasi to really pinpoint what pressures select a dominating microbe within a community, a dominating species within a community, ano siya mahirap siyang i-prove in vitro. So we will always come to probably a truce, no, between the concepts of everything is everywhere and the environment just selects, with the idea of microbes really being globally distributed, no, and we're just not able to capture yung kanilang existence at a particular time. Maybe we can settle for a truce as of the moment, but we expect that selective pressure, or we going back to selection, no, as an important process of microbial community assembly. So we expect those organisms that are highly favored, that are selected to exist on the community. Of course, they will definitely dictate how assembly will proceed, or assembly will not proceed. The last topic, or the last process, is drift. No, drift refers to stochastic changes in the relative abundance of organisms. So you can relate this to the process of dispersal. When we talk about microbial communities, microbial systems, drift demonstrates its contribution usually when selection is weak. So usually if what they call this, yung mga yung environmental pressure, kung hindi nila masyadong nai-induce yung process of um, uh, picking, or allowing uh, select organisms to survive, select microbial organisms to survive in such conditions, when the environment has poor influence on microbial community structure and assembly, drift comes into play. Also, when the alpha diversity is low, so in instances when what do you call this, when specific samples or diversity of specific samples, if it's low, species diversity is low, drift also influences. Kasi let's say, for example, you only have 3 species in a sample, in a microbial community from an environment. If one of the species decides to drift or disperse, go away from that environment, almost 30% na, or 33% nung microbial community nawala na siya. So ang laking effect noon sa succeeding processes. Kung let's say kung totally lahat ng mga species na yon nawala sila or nagdrift sila, will assembly still push through? May or may not. No? Okay, and also drift influences greatly if the total number of community members is small. So essentially, we can say na drift comes into play if diversity level is very low. If diversity of the microbial community is very low, and the environment does not put a lot of pressure yet. No. So remember spatio-temporal ang mga proseso natin sa microbial community assembly. So you don't only factor space, the environment, the space that is occupied by the community, you also factor time. So lagi natin iisipin 'yon. Kaya nga, a lot of things in microbial community assembly a lot of rules are not generalizable externally, because specific environments will always show, most of the time will demonstrate patterns that are unique to them only. Kaya very important na malawak or open-minded tayo pagdating sa microbial community assembly. There are less rules, no, or less external validities that we cling on to when we talk about microbial community assembly. We always have to remember that microbial community assembly, its establishment, its succession, its structure, changes or reshaping will always be married with the status, with the physical chemical status of the environment. Okay? So for further reading, I have a few things that I want you to look at. Some examples of microbial communities. Please go ahead and read some case examples so you'll have an idea. Planktonic microbial assemblies in marine ecosystems, Microbial communities found in hot springs can be found on the water itself or maaring dun sa surrounding terrestrial soil. Also, microbial communities that are associated with wine production. Want you to read that. Wine and cheese production, almost the same, no? Very comparable. So please read on some of these examples of microbial communities as they assemble and how are they proceeding with normal processes or with their normal succession succession processes. Please read about them. Okay, and these are some of our synthesis questions for this module. So that ends our module for factors affecting microbial community assembly and the nature of microbial communities. Okay.